Hi YouTube, my name is Michael Berenger. I'm a medical doctor and a full professor of sports medicine and exercise physiology at Goethe University, Frankfurt, Germany. Welcome back to the Muscle Physiology series. Every movement you make, from lifting a book to smiling, depends on the interaction between two microscopic proteins, actin and myosin. This elegant process is known as the cross-bridge cycle. And in this video, we'll walk through its different molecular stages and uncover how ATP drives them. We'll also explore how muscle structure and mechanical conditions influence contraction, including sarcomere arrangement, titin, and the Hill equation. So let's do this. In our last video, we explored the structure of skeletal muscle, from vesicles down to sarcomeres. Now let's zoom in a little bit further and see what happens at the molecular level. First of all, in the resting state, in the resting skeletal muscle, most myosin heads are detached but energized. What does that mean? They've already hydrolyzed ATP into ADP and inorganic phosphate, and the lever arms are cocked, ready for contraction. But low calcium keeps actin sites blocked by tropomyosin. When calcium levels rise, actin becomes accessible and the cycle begins. And the next step, there is first a weak binding from myosin to actin. So once tropomyosin shifts, the energized myosin head binds weakly to actin. Like a climber testing a handhold, this flexible contact allows adjustment before firm attachment. And structural studies show surface loops in the myosin head help orient it for the actual movement, the so-called power stroke that happens in the next step. And it's the release of inorganic phosphate that converts the weak bond into a strong one and triggers the power stroke. A rapid swing of the lever arm, and new cryo-EM data suggests this swing covers around 93 degrees, generating mechanical force and pulling myosin towards the dead discs. You can think of this as a molecular ratchet. So once phosphate is released, the cleft in the myosin head locks shut, making the stroke irreversible. And to contract a single sarcomere by just 10 micrometers, this cycle must repeat thousands of times. After the power stroke, ADP dissociates, leaving myosin tightly bound to actin in the Rigor state. Without ATP, detachment is impossible, as seen in rigor mortis. Remember, in the resting muscle, most myosin heads are not in rigor, but in the energized, detached state. When a new ATP molecule binds to myosin, this triggers the detachment from actin and resetting the head. This completes the cycle and allows the process to repeat asynchronously across billions of myosin heads. Now let's talk about the classic force-length relationship. Sarcomere length directly affects force output. This fundamental principle was established through the pioneering work of Huxley and colleagues in 1966, who measured how varying sarcomere length alters the number of cross bridges that can form and thus the force a muscle fiber can produce. At very short lengths, the actin filaments overlap excessively, interfering with myosin binding. As the sarcomere stretches towards an optimal range, around 2.1 to 2.2 micrometers, overlap becomes ideal and force peaks. Beyond this range, overlap decreases, and therefore fewer cross bridges form and force declines. This explains the bell-shaped force length curve a foundational concept in muscle physiology since the 1960s. Let's take a step back. How sarcomeres are arranged inside a muscle defines its mechanical performance. The more sarcomeres a muscle has in series, the greater the total shortening distance, range of motion, and contraction speed. The more sarcomeres are arranged in parallel, the larger the cross-sectional area and the more force the muscle can generate. Of course, every muscle combines both layouts. Its architecture, including fiber length and pronation angle, therefore reflects the muscle's specific functional balance between speed, range, and strength. And now let's talk about the Hill equation. Muscle length doesn't just affect filament overlap, it also changes cross-bridge behavior. The force-velocity relationship in muscle was first described by Archibald Vivian Hill in 1938, who formulated a mathematical equation to capture the inverse relationship between load and contraction speed. Decades later, Seo and colleagues showed that this relationship arises from cross-bridge kinetics. At high loads, myosin heads remain bound longer, increasing force but limiting velocity. And at low loads, they cycle more rapidly, favoring speed over force. Vanwyck and colleagues in 2021 observed a related effect with sarcomere length. At longer lengths, myosin detachment slows, sustaining tension. At shorter lengths, detachment is faster, improving speed but reducing the number of strong bounds. In both cases, the underlying principle is the same. Contraction mechanics are shaped by how long myosin stays attached to actin depending on load and length. Finally, let's talk about titin. And titin is more than a spring. Titin is a giant and yes, a spring-like protein spanning from the Z-disc all the way to the M-line. Once seen as passive, it's now recognized as an active player in muscle force. And Tomarka and colleagues in 2020 showed that titin contributes to force even when myosin is blocked. During stretch, titin stiffens and stores elastic energy, which is released during shortening, increasing efficiency without using ATP. Titin also explains history-dependent effects like residual force enhancement and residual force depression. Hahn and colleagues suggest that active stretch stiffens titin, increasing post-stretch force. And in contrast, after shortening, titin slackens and force drops. These results speak in favor of a 3-filament model, rather than a system based solely on actin-myosin interaction. So titin actively contributes to force generation and plays a role in the muscle's mechanical memory. Let's recap. The cross-bridge cycle consists of several distinct phases and it is driven by the energy provided by ATP. Muscle force depends on sarcomere length and arrangement. Titin and strain history add a new layer to the contraction, and cross-bridge kinetics explain how load and length shape force and velocity. In the next episode, we'll explore how muscle activation begins from the arrival of a nerve signal at the neuromuscular junction to the release of calcium inside the muscle fiber. What exactly triggers contraction? Stay tuned. Thanks for watching, and next time you move your finger or take a step, remember, billions of myosin motors made it happen. See you soon. Bye-bye.